everyone, and welcome to today's webinar titled Applications of Multiplex Digital PCR, Monitoring Wastewater for SARS-CoV-2 and Measuring Bacterial Gene Expression. I'm Christy Jewell of Labyrinth, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labyrinth and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit thermofisher.com. Now let's get started. You may participate by submitting as many questions as you like at any time you like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. I now present today's speaker, Joan Slonczewski, Professor of Biology at Kenyon College. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Joan, you may now begin your presentation.
So thank you for that introduction, and I'm excited to be here today describing our experiences with developing wastewater virus monitoring for a small community. So back in 2020, as the pandemic got started, our small village of Gambier in Ohio was interested in how we could use some of the signals available from molecular biology to give information to the community about the pandemic. And you can see here what it looks like in Gambier, Ohio. People had a lot of questions like, should we wear masks? The mayor had a big meeting as to whether there should be a mask mandate for the village. And people wanted to know, should they go to a restaurant? And the information was always very delayed. So there's a dashboard of public cases, but it could be a week or two before you would see the actual results of cases. So wastewater monitoring for virus was something that was being developed by a number of labs in Ohio. The state of Ohio is actually at the forefront of this effort. And so we participated in that. So what we did is we developed a method to monitor shed virus, in particular the RNA from shed RNA viruses from the village of Gambier wastewater treatment plant. And actually the idea of monitoring viruses of viral RNA has been around for 20 years, and there were literature techniques to do it. And this particular virus, there were published probes and PCR amplification probes, that the same ones that are used in clinical testing turned out to be usable in wastewater. So the village of Gambier, the wastewater treatment plant, is actually, it's a small plant, only 100,000 gallons per day, and it's a pretty tight plant. There's not too much infiltration. So it was a good case in a number of ways. Now the village population is small. It happens to include about 2/3 of the population are undergraduate students from Kenyon College who live in residence halls in very close quarters. And so there's the opportunity for rapid spreading of virus there. Now, during the past year, there was a community dashboard that posted the results for active cases, both from Gambier, and then there was also a college dashboard. So we had case data, and the case data are shown in red, the published cases. Of course, we know that there are many cases of SARS-CoV-2 that escape actual appearance with symptoms. So it was of interest to monitor, can we detect the RNA from virus of people that are not infected as well as those who are infected. So what you see here is the results that we were able to obtain with the Thermo Fisher instrument. You can see that the blue curve indicates the log2 RNA copies per liter that we were able to measure, and we can see the progress of that over the last fall and the corresponding data from case reports in red. So the interesting thing is that, as has been shown elsewhere, the Ohio State Monitoring Group and others have shown that wastewater does act as a leading indicator of cases, so that the viral RNA appears in the wastewater about a week ahead of typically case reports. Then once you have a spike in cases, the wastewater RNA levels do tend to lag behind. So it's considered a lagging indicator of case decline. What happens is that people that are still shedding virus are combined with people that are new infections. So if you see the wastewater levels declining about half per week, that's a good indication that virus in general is declining and that you don't have new cases. But the most important thing for people in the village was that you have this leading indicator that you can see that there's an infection spike starting before the cases are actually reported. And that's useful for somebody considering, well, am I going to go out to a restaurant this weekend? Things like that. So what was the technology that we used? And so basically we went and obtained wastewater samples from the influent of the village plant. And so we see here a wastewater sample and a couple of tubes that have salt. The salt rapidly inactivates most pathogens that would be in the wastewater. I should add that we assume, and this all the literature has borne this out, that the SARS-CoV-2 virus is rapidly inactivated in the wastewater prior, that what you're detecting is RNA from inactivated virus. There is not believed to be infectious virus in the wastewater. So we would obtain the samples in the morning from an auto sampler that had cycled every 15 minutes to get an average sampling over the past 24 hours. And we then took it back to the college where my students and I conducted an RNA purification. And the purification method that we use, there are a variety of different tools for purification, but we did the simplest possible purification that we could find, which was from the Berkeley University. It's out there on the internet. And so basically what you do is you first inactivate with NaCl, then you assume that viruses and other pathogens are lysed and that any RNA is removed from particles. You filter that wastewater. Now, the filtration that we used did retain some small particles smaller than 5 micrometers. So there is, in the literature, there's some sense that some of the viral RNA is actually adherent to the particulate matter of wastewater, which is a very complicated fluid. So we're capturing some of that in our prep. Then this is ethanol treated and treated with a buffer that should remove most of the RNA from any particulates and pass through a lysate through the zymosilica spin column. This is a spin column of silica that specifically attaches nucleic acids, and most other stuff pours through. And in the eluate, we took concentrated RNA. The RNA would have been concentrated about 400-fold from the original sample. And this was a process that was simple enough that I could do, and also my undergraduate students, 2 undergraduate students were actually hired to help out doing this. So here is one of them, Bailey Fitzgerald, and here she is setting up to run the absolute Q instrument to measure the amount of viral RNA in the samples. So the assay that we used was assay that was developed by Combinati before they were bought out by Thermo Fisher. So the Combinati 4-plex assay includes 4 different fluors in 4 different TaqMan probes for different primer sets. So the primer sets N1 and N2, those are based on the standard CDC primers that are used in all the swab tests. The bovine coronavirus was for a control coronavirus there was some debate as to the usefulness of that. We eventually decided that we did not require that control, that we felt it was not really relevant to the SARS-CoV-2 virus. But then a very important control is the pepper model virus, which is a fecal model indicator. And so that virus was detected with the Thai floor. So we used those three, and later versions of the probe mix were 3-plex and included just those 3 floors. So in order to perform this, we did use the absolute Q instrument, in which there are 16 wells, and each well for a sample. So for each sample, the final sample volume is to be 9 microliters exactly. So this included the 4x or 3, the 4x, excuse me, the 4-fold concentrated master mix that includes the reverse transcriptase, then 20-fold concentrate of the probe mix, and the RNA sample. And there's room for about 2/3 of the volume consists of the RNA sample. We also ended up, by second semester, we started using a specific probe for a variant, which was the Omicron variant. So we did not do that first semester in the data I showed, but for this current semester, we've been using the Omicron 2-plex probe, which was also developed by Thermo Fisher. And this is based on the spike gene, a particular mutation, the spike gene in which glycine is replaced by a different amino acid, and by aspartate. And so there are 2 probes that compare the Omicron variant result with the standard reference, which would have been the Delta variant at that time. And it turned out that both probes were able to work under the same settings. So I should also mention the RNA sample that we used. We found by experiment that the RNA actually required some dilution because the RNA has some PCR inhibitors in it, and this has been generally found with wastewater monitoring. So we experimented with dilutions, and we found that if you diluted 5-fold or 10-fold, that your signal actually increased. So we ended up routinely diluting the sample in TEA buffer before including it. So here's what the instrument readout looks like. So for a typical run, this would take 2 columns of the 16-well plate, and we would have 2 independent samples of the wastewater from that day. And for each sample in column 1, we would assay the 5-fold dilution and the 10-fold dilution of the wastewater. And then in column 2, we did the same thing, but used the Omicron probe test instead. The cycle parameters you see are pretty common parameters, and we found that the 55-degree annealing step is actually pretty important because the lower the temperature actually improves some of the separation of the signal cloud of reactions from the background. And sometimes using a lower temperature than that of the recommendation for the TACMAN probes was a good thing. And we found that 55 degrees worked well for both of our probe sets. So here's a close look at what the data look like for individual samples. So you can see, for example, for the N1 probe, that the black square represents the 20,000 plus reaction wells, and each dot, it represents one reaction where there was a PCR amplification. And we assume that each of those reactions represents one molecule of viral RNA from the original sample. So that's a pretty powerful technique where you can be counting individual RNA molecules out of your sample. And in the upper diagrams, you can see the Poisson distribution represented. So the positive signal, the positive reaction wells are indicated above, and then below the threshold, you see the large bulk of the 20,000 wells did not react. And you see a similar picture for the N2 probe, the hex 4, and then the ppMOV is the control saying that yes, we have significant fecal wastewater content here. And you could tell that worked because when students went away for vacation, students were actually about 2/3 of the population. So the ppMOV fecal probe went way, way down when the students were out of town. So here we see an overall picture of the results from a couple of days that we looked at. We also compared a recent day's results in January 12th, with a frozen sample from September. So if we look at first at the left half of the slide, we see for January, we see the results of the 3-plex probe, and we can see that for both the hex and the FAM, okay, the blue and the green, you can see that there is definite signal there. So there was quite a bit of wastewater virus on those dates, and that was when students had just came back and we had a spike of cases shortly thereafter. And so we can also see the control as well. Now on the right-hand side, we see the results of the Omicron probe set compared with the older September date. And so the blue floor is the one that detects the Omicron specific probe, and you can see that on January 12th, that probe shows specific reactions, whereas the control from the older virus variant shows nothing. And by contrast, the frozen September sample shows positive results for the Delta variant and nothing for the Omicron variant. So this shows that, in fact, we were very precisely able to see. And there was another day also where it was mixed, okay, where it looked like there was some Delta left and some Omicron coming in. So you really can see novel variants just by shifting your probe. In terms of controls, we use two kinds of controls. One is the fecal control, so that, for instance, in this well, you can see a little bit of a couple of fecal counts that shouldn't be there. So I did talk with the students afterward about making sure that the loading was good and how to keep pipettes uncontaminated. The other kind of control is your wastewater good. So the fact that we always found actually increasing counts for the 10-fold dilution versus the 5-fold dilution means, yes, that's looking like real wastewater because you're seeing the result of the inhibitors as well. So we relied on internal self-controls as well as the presence of the fecal control in the 3-plex probe. So for an overall summary of what we would have seen on those days, so we had essentially 4 replicates, technical replicates, looking at the N1 and N2 probe sets. It was pretty consistent that the N2 probe set showed higher numbers, and we generally use that one for our community reporting. You can also see it was clear that we were seeing Omicron on the recent date and Delta on the older date. So at this point, I would say that we are continuing to use the wastewater data, that it continues to be considered to be of use by the community. In fact, what we did is we posted these data on the timeline graphs that you saw earlier, and people are saying, well, it's good to have these data, especially as the community opens up and not everybody's wearing a mask anymore and they don't report all the cases, community members consider this really helpful to have and have been able to understand at the level that is presented. We also have an email list where we present explanations of it. In addition, it's really important to know that it's fairly straightforward and easy to do this kind of monitoring. I have 2 undergrads who conduct the analysis, and it's drawn interest in further kinds of research using this technology. So what we're doing now at Kenyon is we're applying to other questions. For example, drug resistance, and how do we deal with drug resistance and the effectiveness of antibiotics and multi-drug resistance genes. In fact, some reports are coming out showing that actually there will be a lot more fatalities and morbidity due to drug-resistant bacteria in the next few years than just something like COVID. This is really a pressing problem. In fact, it's been amplified in the hospitals because as more people go to the hospitals with COVID, then the hospitals are not following up on drug resistance as well as they should. So we're looking at research questions related to drug-resistant bacteria. And a particular question we're studying in our lab is how do oral pharmaceuticals and as well as phytochemicals in our foods affect the bacterial expression of drug resistance genes? So when you ingest food, you have all kinds of phytochemicals, including organic acids, and we have a hypothesis that these organic acids affect the relative fitness of the multi-drug resistance phenotypes in bacteria, and therefore can alter the gut microbiome and make you more or less drug resistant in your microbiome. So we're interested in looking at multi-drug efflux pumps, which are driven primarily by the proton potential, and the proton potential is affected by organic acids that leak hydrogen ions into the cell. So we're looking at the specific question of how expression of these drug pumps is affected by various compounds. And so this is an approach where we can use the same configured assay, this 4-plex assay, using 4 different fluorids and 4 different TaqMan probes. We can use this to simultaneously assay 3 different multi-drug pump expressions, as well as the RPOB control, that's the RNA polymerase enzyme expression as a control. And so we're just developing this application now, but we find that the data actually look very similar to the wastewater data. We designed specific probes. In this case, we had TaqMan probes designed by a company that's expert in that. And I must say that even on our first try, we got 4 TaqMan probes that work well, and they work actually following the same assay configuration that was used for the wastewater samples. So we can use this assay to screen RNA preps from bacteria cultured under various conditions of pH and drug exposure, and then we can screen many different conditions with this 4-plex assay, and then identify the most promising conditions for later RNA-seq follow-up. So we're finding the absolute Q and the 4-plex assays to be a really exciting application in basic research as well as for community wastewater monitoring. Thank you, and I look forward to questions in the question and answer period. I would like to acknowledge all the, excuse me? So I would like to acknowledge all the support that we've received from the Ohio Coronavirus Wastewater Monitoring Network. We received a sub-award from the Ohio Water Resources Center and the Ohio Department of Health. And the Village of Gambier was very supportive and encouraging. We also received funds from Kenyon College, Office of the Provost. And I would especially like to thank my students, Bailey Fitzgerald and Langyang Lou for their work in this wastewater monitoring.
Thank you, Joan, for that informative presentation. We will now start the live Q&A portion of our webinar. If you have a question you'd like to ask, please do so now. Just click on that Ask a Question box and click Submit. We will answer as many of your questions as we have time for. So, Joan, we have some great questions coming in. I'm going to start with this one. Why did you select digital PCR as a technology to quantify nucleic acids for these applications?
Well, digital PCR was recommended by some of the Ohio State professors with the wastewater monitoring network, and I had already previously done wastewater monitoring by sending the samples out to the Lumen Ultra company, but that took a week's turnaround. And so I was impressed by the fact that this technique, the digital PCR, could count individual molecules of RNA, and that it required a small number of controls. And so it looked like something that would be easily doable, something that could be learned to do easily and could give rapid results.
Thank you. Our next question. What were the impacts with the data collected for this study on the community? And do you see this method being used in the future as we hopefully transition into an endemic phase of this pandemic?
Well, the most important impact of the data collection was the confidence of the village community and the Kenyon community, of many Kenyon employees as well as students, and the village saw this as providing a picture of the pandemic for them and gave them more confidence in the overall kinds of data that they were getting. And it helped enforce the village mask mandate to say, well, even if there isn't a case reported today, the wastewater shows there's still virus present in the community, things like that. So in the future, there's a lot of interest in wastewater monitoring for a whole range of viruses. It can be used to monitor influenza viruses and enteric viruses, noroviruses as well. So I think that in the future that there will be a lot more of this kind of technology.
And Joan, what other environmental testing applications do you see digital PCR being useful for?
So digital PCR is useful for all kinds of environmental science, for measuring small quantities of RNA and DNA molecules. So I think that it can also be used to measure viruses in natural water bodies. We do microbiome analysis of streams and ponds, for example.
Thank you. And what are the advantages of using multiplex digital PCR over other methods to measure gene expression?
Well, the biggest advantage is that it can be done rapidly on many samples, and you get data back soon, and it has the ability to measure single molecules. So the amounts of RNA that you use are tiny. We found that the amounts of RNA that we need for our bacterial preps, for example, it might be 1/1000th of an RNA prep is what you need for a run. And so the fact that you need such small amounts of RNA is really great for research applications. There are now RNA micropreps that have been devised that can obtain very small amounts of RNA from a large range of samples, and you couldn't have afforded to do this before. So the fact that you can get many different samples from different conditions is really good. And the fact that you can get this information, and then it also serves as a cross-check for the RNA-seq applications, because it's a different technique that provides information in a different way compared to the RNA-seq approach to gene expression.
Joan, you've given us so much information today. We do have time for one final question, and we'd love to know, what additional information do you gain from using this technology?
So the main additional information that we gain is that we can target specific genes in a system and ask questions about those specific genes over a large number of conditions, say a range of concentrations of an exposure pharmaceutical. So it's a technique that can enable us to target specific genes and to detect the smallest quantity of expression of them. And of course, you can also use the technique to query DNA samples as well, so that I have colleagues that are doing mapping of DNA, long distance mapping of DNA in chromosomes, and you can use the technique for that as well.
Thank you, Joan. Now, do you have any final comments for our audience today?
Well, I would just like to say that if you know any young people considering colleges who want to do science research, that Kenyon College is a great place to send them, that they will get a real science education and get participation in state-of-the-art research and get to use great instruments like the Absolute Q in their research.
Thank you again to our speaker, Joan Slonczewski, for her time today and for her important research. We'd also like to thank LabRoots and our sponsor, Thermo Fisher, for underwriting today's educational webcast. Now, before we go, I do want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, they will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you again for joining us. Have a great day.